Hey folks, this is Bryce, and this presentation is on the loop-shaped minimally invasive cytologic brush. So to start off with some data on pancreatic cancer and pancreatic cysts. Pancreatic cancer is extremely lethal. If you have the disease, there is only a 5 to 10% chance that you are going to be alive after 5 years. That's what the 5-year survival rate is. However, if you are diagnosed early enough, there is a 10% chance that you will actually live. Unfortunately, this doesn't happen very often because catching tumors is very hard in the early stages. That's because they are small, they're hard to find for biopsies, they're hard to image, and there's no single conclusive blood test. Pancreatic cysts themselves are even harder to catch. 1/5 of all biopsies on this tissue type fail because of lack of sufficient cell count using the current standard method. And up to 1/3 of all pancreas resections, which are removals of portions of the pancreas because of malignant tissue, occur because of cancerous cystic tissue. Therefore, if we could improve the ability to diagnose pancreatic cystic tissue, we could save a large minority of patients if that diagnostic procedure is applied early enough. So one of the ways of imaging pancreatic cysts and the pancreas in general for diagnostic purposes is endoscopic ultrasound, or EUS. In addition to having the potential to perform imaging, EUS can also be used in tandem with a technique called fine needle aspiration as a technology for biopsies of pancreatic cysts. So the same technology applies. The endoscope goes down through the mouth, down into the duodenum, and then a needle, which comes out of the tip of the endoscope, pokes through the duodenal wall into the cystic tumor, and then aspiration of the cystic tumor cells takes place, which is vacuum suction through the needle and back up the endoscope. So EUS FNA is a very useful tool. However, as I mentioned before, 1/5 of biopsy attempts of pancreatic cysts fail using EUS FNA. And the reason that they fail results from the fact that most of the cells within a cyst can be found in and on its inner epithelial wall, and they themselves are not suspended in the cystic fluid. So when aspiration of the cystic fluid is performed, very little sample is actually obtained. Accordingly, a technique called through-the-needle cytologic brushing was invented. In this technique, a brushing apparatus is deployed through the needle at the end of the endoscope. And when the brush is rotated, it abrades the walls of the pancreatic cyst, scraping cells off of that wall and into the cystic fluid for aspiration of a higher cell count. Unfortunately, the pioneering technology for this through-the-needle method, despite being more effective than EUS FNA alone, posed a bleed risk to the patients and was discontinued. Accordingly, the loop-shaped minimally invasive brush was invented as a gentler, effective alternative to the echo brush. In the pilot study by Marquez et al., the LSMIB was able to collect up to 174 times more cell content from ex vivo porcine cyst models than EUS FNA alone. The innovation of this brush is that it's made of a nitinol wire, which is a nickel titanium alloy. This alloy has what are known as shape memory properties. These properties allow nitinol wire to be heat trained into a certain shape. And after that heat training, when sufficient cooling is applied, the wire will be extremely malleable and compressible. However, when heat is reapplied to the wire, it will undergo a phase change and conform back into its original shape. So what that means in the context of the LSMIB is that when the brush loop wire is inside of the needle before deployment into the cyst, it is flexible and compressed. But when the wire enters the warmer environment of the cystic cavity, it springs back into a circular shape and conforms to the spheroidal shape of the cystic cavity. Not only does the nitinol wire have a useful shape, but it is almost 2.5 times more elastic than stainless steel wire, which is the industry standard for cytologic brushes, making it a gentler alternative. Understanding the specifications and the mode of action of the LSMIB, we can now look into the necessary application pathway and approval process. Given that the LSMIB falls under the FDA's category of device called the endoscopic cytologic brush, a classification level and a common approval pathway already exist. Endoscopic cytologic brushes are considered Class 2 or moderate risk devices. This makes them eligible for a 510(k) approval pathway. Additionally, devices in this class also qualify for a third-party 510(k) approval route. Using the third-party approval route, a 510(k) application is sent to a third-party review organization, or RO. The RO then reviews that 510(k) to determine if the device in question is going to be as safe and efficacious as its market predicate, where a predicate is a sponsor-chosen, sufficiently similar previously released device that is legal to sell in the United States. If the new device is shown to be at least as safe and efficacious as the market predicate, it is considered substantially equivalent. The predicate chosen for the LSMIB is called the ST Cytology Brush by Hobbs Medical. This brush consists of the same basic three components: a handle connected to a guide wire and a guide wire connected to a brush. Both the device and the predicate undergo the same mechanical operations as well. They are run through the endoscope, rotated, and used to brush some sort of epithelial surface. The principal difference is that the ST cytology brush is too thick for EUS FNA. Accordingly, safety and efficacy data comparing the LSMIB to the predicate to determine substantial equivalency needs to be performed first in the GI tract alone. Once the LSMIB has been shown to be safe and efficacious in the GI tract, additional data will need to be collected by the sponsor comparing the LSMIB to EUS FNA alone in pancreatic cysts. Given that EUS FNA is a pre-existing technology, negative control data should already exist. In totality, the 3P510(k) application needs to include final draft labeling, the technology specifications, performance data that was just collected, including in vivo cyst and ex vivo cyst model data, as well as comparisons to the predicate, sterilization information, and any FDA guidance documents that may exist. In total, it should only take a little under 2 years for the LSMIB to begin production.